Hello, everyone. Welcome to Chromosomes and DNA webinar. Thank you for joining us for this session hosted by 3D Molecular Designs and the MSOE Center for Biomolecular Modeling. I'm Heather Ryan, and I'll be your host for the series. Before we get started tonight, I would like to remind you about our free coronavirus video series. This 4-part series contains hours of videos, which are broken up into short segments, but they're geared for your students. These videos are paired with paper modeling activities, molecular visualization activities, and links to other resources. Thousands of people have already watched the videos. The series pairs well with the CBM's antibody maps module and our new plastic coronavirus models. So Chris Chow is going to be leading us through the Chromosome Student Modeling Pack and the Chromosome Connections Kit tonight. We will answer all questions that you have for Chris before the break because she has to get back to parent-teacher conferences tonight. After the break, Tim will focus on epigenetics and we will do the regular questions and answers at the end. So now I have the pleasure of introducing Chris Chow. Chris holds a bachelor's in bioengineering and a master's in curriculum and instruction from the University of Illinois at Urbana-Champaign. She's been a science teacher for 20 years and enjoys facilitating workshops for science teachers in her district, at national conferences, and now webinars. She received the 2014 Spotlight Award for Excellence in Teaching from the St. Vrain Valley Education Foundation and the 2016 Outstanding Biology Teacher Award in Colorado from NABT. She currently teaches AP Biology, Biotechnology, and coordinates the Medical and Bioscience Academy program at Longmont High School in Colorado. And now, Chris will begin with making connections between DNA and chromosomes.
Hello, everybody. It's great to be here with you. I'm taking just a little break from my parent-teacher conferences tonight and to talk to you about the chromosomes and DNA modeling kits offered by 3D Molecular Designs. So I'd love to find out from participants where you live and what grades and subjects you teach. So if you wouldn't mind, go ahead and type that in the chat so that we can introduce each other. All right. All across from West Virginia and Connecticut and Alabama. Fantastic. All the way in Hawaii. Okay. Lenore. Fantastic. So welcome, everybody. So some of our goals tonight are to model chromosome structure and anatomy. I hope you all received your student modeling packs because we're going to have you break those out tonight and actually model with us. We're going to use those kits to compare and contrast mitosis and meiosis, model mechanisms that contribute to genetic variation, and kind of a bonus at the end is show how you can use these kits in Punnett squares to make the connections between the molecular level with chromosomes to traits. So I thought I'd share with you a few of my favorite photos. These are students that I had in my AP biology class the year before COVID hit. So this was fall of 2019. And as you can see, I love using any chance that I can get to be able to get kids with their hands on a variety of models, notably the 3D Molecular Designs ones, because one of the important science practices is to be able to utilize and discuss and represent scientific ideas and concepts using models. Kids are really engaged in class when they have a chance to do this. And I especially love being able to elicit student understandings and some misconceptions. They enjoy chatting with each other in normal times and having those conversations. And I love circulating around the room, seeing them actually move physical pieces and have discussions. So this kit is one of my favorites. And you can see that these are my students using them, and I organize them by I purchased like the large classroom sets. So I have one large kit per table group, and I organize them on these little trays that you can see there. And so you can see some of my students kind of working through crossing over in meiosis there. So we'll get to that tonight as well. Okay, so here's what you need to make sure that you've got ready is your chromosome student modeling pack. For some of you, if you received the email earlier, we wanted you to do a little bit of advanced work so that you could not worry about scrambling and getting everything ready. But if you are just now opening up your plastic pack, if you've had that busy week so far, no worries. You can kind of assemble some of these things later as well, and just do the best you can to follow along with us. So there is some cutting and gluing and taping involved. So you may want to attach some of the foam pieces onto the scaling graphics that as you see there. And so I want to go ahead and give you about 2 minutes. This is probably not enough time for everyone, but just kind of get you started with connecting some of our foam pieces. Note that there's some nucleotides in the middle between the scaling graphics. Don't worry about the sequence right there, just kind of put them in there with their base pairs because we want to take a look at chromosomes at different levels. So give you a couple of minutes there to assemble your chromosomes that you're going to see on the screen here. All right, and so as some of you guys are working, I did want to share just a personal anecdote. I purchased 55 sets of these student modeling kits in August and actually sent them home with all of my AP biology students. So it was really nice that during the first quarter of the semester, fall semester, all my students were at home. So I was able to do this modeling with them. They really liked having these in their hands at home. I don't plan on collecting them all back and figuring out how to sanitize them all, but they can be, just so you know, if you're looking for kits that can be sanitized, they can definitely be like dipped in a low bleach solution and be handled between classes or different kids. All right, I am not a Zoom user. My school uses WebEx, so we'll see how this goes, guys. It's all about being flexible as we're all learning. Okay, so going back here. So this graphic shows the organization of eukaryotic chromosomes, and you can tell that it's eukaryotic in that bottom picture because it's a linear molecule. And so you can see that we're zoomed in at the very top with the DNA double helix, as we're used to seeing with the individual nucleotides shown with the base pairing. And then we're zooming out and seeing the organization of those strands of DNA wrapped around the histone proteins. And then there's further coiling and condensing as cells are getting ready to divide. So you're going to see that chromatin condense into chromosome visible physical structures, which are at the bottom. So this is different representations of DNA zoomed in close to being zoomed out. So take a look at your model that you created with your foam pieces. Can you identify all these different components in that graphic? So multiple ways of showing DNA at several different levels, from the nucleotide level with A's and T's and C's and G's base pairing, to the scaling part of the graphic, which is that flat piece. And you can be able to see the organization of DNA wrapped around histone proteins, and then the coiling and condensing to the large foam pieces, like the one seen at the bottom of the screen there, where you have what traditionally, you might use other pieces like pipe cleaners or pieces of paper to show movement of large pieces of chromosomes. So this is really handy because it helps students to see DNA at multiple levels to make the connections between nucleotides to the large chromosome structures. Okay, so a couple of things to show you that are differences. On the right is the student modeling kit. They get kind of a baby version of the nucleotides and fewer chromosomes. And the larger version on the left is the classroom set, the chromosome connections kit. And you can see the nucleotides are either all red or all blue to show the maternal and paternal chromosomes. You can do, I think, 3 pairs of chromosomes with the large classroom set, and then 2 pairs of chromosomes with the student modeling kit. Okay, so we're going to zoom out and go to the larger chromosome level. You're going to need to remove some of the chromosome foam pieces off of your scaling graphic in order to create these 4 chromosomes shown. So we're going to give you 2 minutes for that. Okay, so hopefully you've got those chromosomes made. They're still shown on this image here. Now let's talk about the different parts and components that are represented by this model. So the little round ends are the telomeres, and the in terms of the colors, the blue represents paternal chromosomes that come from sperm cells, and the red represents maternal chromosomes. And you'll notice there's a 2, 1 blue and 1 red of each length of chromosome. And so those are your homologous chromosomes. And then of course that center part that looks kind of pinched in is the centromere, the region where later on we're going to be able to talk about how spindle fibers can attach to that section of the chromosomes during cell division. So this is kind of handy for students to have it in front of them, and you can ask them to point to and identify these different parts of the chromosome. Okay, so this representation is a human karyogram, and it allows us to be able to organize chromosomes based on their size and centromere position. And so you can see one of each type in this karyogram. But of course, as humans, we are diploid, so we have 2 copies of every type of chromosome, and you can see this shown here. So these are stained chromosomes and their banding patterns. And this is an actual photo of a karyotype, so of showing you all the different chromosomes lined up. And keep in mind, this photo is showing you metaphase chromosomes. So each individual piece that you're seeing there of those chromosomes, for example, number 1, they're already duplicated. So if you look really closely, you can see those are duplicated chromosomes. That's something that's really important to note, okay? All right. So with that, as we talk to students about how do we get this picture of the chromosomes, it's important to talk about the cell cycle and kind of put that into context for students, that the cell cycle describes the life cycle of a cell from where it's in G1 in its regular growth phase to all the way to S, where the chromosomes get duplicated or replicated in DNA synthesis. And then as the cell prepares to divide, it goes through G2 and then through M phase of the mitotic phase. And so in the middle there, you have metaphase, where the chromosomes are all lined up on the metaphase plate. That's an ideal time when all your chromosomes are fully condensed. Before they are separated, you're going to see that the karyotype is going to be taken. The picture of the chromosomes are going to be taken right there, okay? Okay, now we're going to go through DNA replication and replicate your chromosomes. So for every chromosome you have, you're going to make identical copies. And I'm going to give you 2 minutes to do this as well. So there's a couple of questions coming in. From Christina, do I let my students assemble all these parts, or do I assemble them prior to handing them out? If the students have them at home, they're going to be assembling them as I talk them through it. So they do it themselves. With the classroom, with the classroom kit, let me go ahead and pause that there. With the classroom kit, I do have my student aides kind of punch everything out and assemble them. So they do that. And then so that they're ready for them, and we don't spend so much time in class actually assembling them. They can just get right down to identifying parts and modeling the processes of mitosis and meiosis. So it looks like Heather's talked to Phyllis that they might have to ship you some additional centromeres there. And then from someone in the Q&A, which foam piece represents the centromere? The centromere is the center part that looks pinched. So it looks like it kind of has this dent that goes in. Okay. So I might have, okay, so with the student kits, I think it's possible we might only have 2 pairs. I think we may have to confirm that with how many parts are in there. My understanding was there should be 2 pairs to be able to do the replication there.
I'll check on that, Chris. And oh, I'm having some interesting lighting. Okay, I'll switch to this.
Okay. All right. So the other thing was, you know, if your students are missing some, then you could just have them sort of draw and trace it out and color in and kind of represent that as well, if there's not enough of those centromere parts until you get your shipment of replacement centromeres. So Heather's got on the screen right there, and she's got, so for the students at home, they won't have access to these connectors, which are the kinetochores and the spindle fibers. These are in a classroom kit. And so a great alternative at home is just to use string or dental floss of some kind to represent the connecting of the centromeres to the spindle fibers. So Heather's got that on camera. Okay, so now go ahead, and you're gonna need some space on that table in front of you to model the process of mitosis. So you can follow along with this picture, or you know, kind of talk yourself through it. Oh, I see. Carrie said that you have enough of those centromeres in your student modeling pack, but you had to disassemble them. That's correct. Yeah. So the one at the beginning, you had to take apart to be able to make the full chromosomes. Thanks for making that clarification there. Okay. All right, so Heather's going ahead, and she's got metaphase right there, where individual chromosomes, these chromosomes that are duplicated, are lined up on the metaphase plate. And then her string spindle fibers are going to help guide those individual chromatids to the 2 poles. Okay. And then, of course, so that's anaphase, and then telophase is the 2 brand new cells are almost there. Okay. I see Phyllis's comment. No problem. This is good for you to experience this, because from the student lens, then you can be ready for those things that your students are going to encounter as well. All right. Okay, so now one thing I forgot to mention is when I use terminology with my students, I will make sure that I hold up one of these chromosomes and make sure they repeat after me. This is a chromosome, and this is a chromosome duplicated. So they have a really hard time distinguishing between chromosome, and then these are sister chromatids, and but a chromosome duplicated. So I also have them act it out with their bodies. So they'll you know, go like this and go, I'm a chromosome, and then put their arms and legs out, kind of like doing jumping jacks. I'm a chromosome replicated or duplicated. And so gets our blood pumping. You do jumping jacks in the classroom. All right, so Heather's going to, or Chris is going to play this little animation here. So that's the classroom set that you're seeing in this animation, where there's 3 pairs of chromosomes. And of course, those yellow parts are the kinetochores that connect to the centromere, and then the spindle fibers are attached to them to help guide the chromosomes to the ends. Okay. All right, so with your students, you can discuss a lot of questions, like asking them to identify how many chromosomes before mitosis, how many chromosomes after, how do the daughter cells compare to the parent cells. You can go over the terminology with chromatids, chromosomes, sister chromatids. So that's a lot of confusing terminology with a lot of words that start with C. And so having them with these models in hand and having them show you or say things out loud or to each other if they're pairing up and talking to each other is really helpful with this kit. Okay, so the next slide, I just wanted to get kind of feedback from you. How do you think this activity might be used in your classroom? Where do you think your students are going to get those aha moments? And maybe for you, having the student lens on and maybe using this kit for the first time yourself, how do you think this could be helpful for your students in learning mitosis or parts of the chromosome? Okay, got some feedback from Leslie. She says that already this semester you've used it, it was great. Okay, thank you. Okay, Susan, it's meiotic recombination. Okay, great. And Eric mentions a really great strategy or tool that students can use to demonstrate their understanding is stop motion animation. Absolutely. Great. Okay, these are, oh, Eric is saying hard to recombine socks. I totally agree. I had created felt chromosomes and can't do that. And I also used a card sorting activity with playing cards to demonstrate mitosis and meiosis. And of course, I had to ask the students, well, when we're doing crossing over, I can't have you rip my cards apart and re-glue them together. So, okay. All right, thank you for the feedback there. Okay, so we're going to move on from mitosis, which is making clones or identical daughter cells, to making sex cells or gametes through the process of meiosis. Okay, so we're going to, the next slide. We're going to walk through the steps of meiosis. And this is where the foam kit in this next few steps that we're going to do together, this is where the foam kit is really helpful and beats socks or pipe cleaners or even the pop beads, because these are way easier to handle. My fingers have really hurt in using those pop beads that some folks have. So these really work well. Okay, so in that graphic, what you should do now is, so if we could go back, Chris, one more. Okay, so notice G1 is going through the stages of the cell cycle now. G1, you have individual chromosomes, okay? S phase, that they are replicated. So DNA synthesis occurs there, so then you can have that phase happening. So hopefully you're following along there. Okay, all right. And then we're going to get into prophase 1. So as these homologous chromosomes are now going to pair up, this is where it gets complex, where someone was mentioning meiotic recombination. So this is showing you a graphic of how these the homologous chromosomes come together, pair up, and they actually will join together, and then there will be some double-stranded breaks that occur. And so it's a pretty complex process, and we're going to model in a very simplistic way, but for maybe some of those of you who teach at the college level or have really advanced students who want to know more, I think the next graphic will help to illustrate that. So this is the classroom kit with all of those nucleotides that are all red or all blue to show maternal or paternal chromosomes. And you can see through this animation, which I think is fantastic. You play it one more time, maybe, to see where there's that meiotic recombination. There's the, okay, there we go. There's the holiday junctions that were created, some double-stranded breaks, and crossing over occurred right there. And so the real key part of all this is not necessarily that crossing over occurred. That's a bonus part of this process. What's really happening is you're making sure that those homologous chromosomes are pairing up so that they can be lined up properly at the metaphase plate during metaphase. Okay, so here we go. Take your homologous chromosomes, pair them up. So that is synapsis, where the tetrads form, and they're called tetrads because there are 4 chromatids there. So they form a tetrad. And this graphic is still not awesome, and here's why. You're seeing this in 2D, where you have these homologous, I keep saying the wrong thing, homologous chromosomes are side by side. But if you imagine in 3D space, as Heather's got on the camera there, you can see that you can stack them up together, and then doing the crossing over, you can see where they can be on top of each other and cross over like as she's showing you in the camera. Okay, so most chromosomes will cross over from 1 to 3 places. There's regions along the DNA that are places where that will tend to have meiotic recombination occur. So those sequences vary between species. And you could see that there's swapping of parts. Okay, so this is where this particular kit trumps the socks, because you can actually remove and detach pieces and swap them there. So go ahead and do that with yours, and then model the rest of the parts of meiosis there with anaphase 1, telophase 1, and cytokinesis. So this is where our students can kind of see the differences between homologous chromosomes, pairs separating, versus individual sister chromatids. And this is where I go around the room, probably spend more time watching kids move their chromosomes for this particular process, because you can see right away whether they understand homologous chromosomes, the pair separating or chromatid separating. And you can talk about whether cells are diploid or haploid at certain stages, and have them actually count the number of chromosomes that are in there. So this is where the model is really useful. And I have black tables in my classroom, so you can use string to mimic, the nuclear membrane, or use string to mimic, the whole cells, or you could use those table markers to be able to, show the divisions of, where the cells divide out, or spindle fibers and whatnot. Okay. I love that there's all these questions and comments, recommendations in the chat. So it feels like you guys are connecting, which is wonderful. A lot of great ideas. Okay. Jennifer, is it Brew, asked about holiday junctions. That I cannot answer specifically. They basically are the locations where there is the connection between the homologous chromosomes, and there's the junction at which there's that double-stranded breaks occur. So perhaps that might be a question for Tim Herman later on, for him to explain a little bit more about those holiday junctions. So here is an animation showing you from the classroom kit 3 pairs of chromosomes and going through meiosis 1 and meiosis 2. All right, so again, you can have your students do stop motion videos, animation videos. And so having these chromosomes out will be great for your students to be able to talk about comparing the number of chromosomes in parent cells and daughter cells, whether it's through meiosis 1 or meiosis 2. Okay, and to compare similarities and differences between mitosis and meiosis. Okay, so this is just for you guys to kind of think about and talk about with your students, is sources of genetic variation. So we have the crossing over, we have the alignment of homologous chromosomes, random alignment, and then independent assortment happening. And then of course, when sperm and egg recombine together. All right. So I think there's a lot in the chat about options and ways to use this kit, or ideas with modeling. So I'm going to continue on with this last piece in the last 5 minutes here, and just kind of talk about how you can actually use this kit with your students in Punnett squares. So I like to have my students play the genetic counselor role, where a young couple comes to you. and they are the Smiths, and they would like to have a family, but there's a family history of a genetic condition that they are concerned about. So we bring in the example of the HBB gene, which codes for the beta globin protein, 2 of the subunits of hemoglobin, which is found in red blood cells. And so that carries our oxygen. You've got that heme group in the middle there. And you can talk to your students about that the HBB gene is located on chromosome 11, so making that connection with genes and chromosomes. And when you zoom into chromosome 11, the location of the HBB gene is on that short arm in that locus that you're seeing there. So helping students to bridge those connections. Now, though, when we talk about alleles or variants of a particular gene, we want to zoom in to the nucleotide level. So you can see here kind of at one locus where there is a difference, a SNP. So you have that SNP right there. And then you can see that the difference is if you're seeing that yellow highlighted area, that in the normal variation of the HBB gene, you have a T. And in the mutant version, you have an A in that location. So that means in transcription, instead of getting GAG, you get a GUG, where in translation, you end up having, instead of glutamic acid, you get valine. And so let's take a look at physically what's happening with that protein. And what's going to happen is, because glutamic acid is hydrophilic, the folding allows you to create that nice roundy shape of normal hemoglobin protein. But when you have the valine, valine is hydrophobic, and that creates a hydrophobic pocket that kind of sticks out. And so it's a kind of a sticky region on the edge of that hemoglobin molecule, and that causes nearby abnormal hemoglobin molecules, also with that little sticky part, to start to clump together. And so you create these long chains of these abnormal hemoglobin proteins, which you can see on the right side. So phenotypically, what that happens to the cell is, and we're going to go ahead and skip to, actually not, sorry. Okay, so we're going to head back there. So that's what's happening with the normal versus the mutant versions of the protein. And so you can have your students build using the nucleotides, the 2 variants, and you can bring in more terminology, calling them alleles. You can look at the single nucleotide polymorphisms, or calling them SNPs. And so you've got normal beta globin protein SNP on the left, so we call that hemoglobin A, and the abnormal version is hemoglobin S, okay? All right, and then connecting that with chromosomes, you can think about if a person inherits 2 copies of that HBB gene, what possible combinations could they inherit if your 2 options are hemoglobin A or hemoglobin S at that particular locus? And you can see that you could be homozygous for the hemoglobin A, or heterozygous or homozygous for the hemoglobin S. And so as a result, depending on which alleles you inherit in your genotype, if you have 2 copies of the normal variant, you have the normal phenotype. However, if you get 2 copies of the abnormal protein, I'm going to go ahead and, so there's a little typo on there. Sickle cell trait and sorry, never mind. The H's are not correct on there. So it should be homozygous, little h down below. Yeah. So the pictures are correct. I have a little typo right there. So sickle cell disease down below would be a little h, and you get 2 copies of the abnormal HBB gene. And so that's going to result in some problems. But if you carry 1 copy of the normal and 1 copy of the sickle version, you get the sickle cell trait. So on the outside, you appear to have a normal phenotype, but there can be some issues that crop up. Okay, so knowing that, you're going to go to the couple and talk to them about, well, this is what's happening, is that abnormal clumping or these strings of proteins that are formed causes the misshaping of these cells. So instead of nice round red blood cells, you get the sickle shaped. And in terms of transporting materials, oxygen through the body, you're going to have problems in those capillaries where they cannot go through nice and in a single file line. So they're going to get jammed up and cause blood clots and pain and problems. So the Smiths come to you and they get genetic testing done, and we find out that they are both carriers of the HBB, the mutant version of the allele. And so you can talk to them about, with your students, you can have them model these particular genotypes, and then you can use the large Punnett square and have them place the nucleotide versions of these alleles onto that placemat, and then have them bring them down and show what combinations could be created if Mr. Smith donated this version versus Mrs. Smith donated this version. So you can see the possible outcomes of what their children might inherit. So that's one way to connect the nucleotides with genotypes, and then also phenotype with what kind of outcome they have. So you could have your students model that, take pictures of them to record that, to talk about that. You won't have enough nucleotides in your kit to model all of them within it. They're going to have to move them in there. Okay, so the last part is having students think about how will the child be affected and what will the chances their child will have sickle cell disease. So you can kind of go through that aspect in the bioethics of genetic counseling, and in informing the couple and educating them on what it means to have sickle cell anemia and some options for their family. All right, so that's just kind of a sort of a bonus there to kind of make those connections between nucleotides and genes and chromosomes with your students.
Great, thank you, Chris. Oh, I forgot to fix my light. I'll have to do that in just a second. So wonderful. We do have a question that came up. And Kasha was curious about the graphic that shows the locus. She's wondering where you might have found that, because her copy that she found before wasn't working, or she couldn't access it anymore. And if you have other questions, please post them either in the chat or the Q&A, and we'll try to address things. I'm going to fix my light.
Okay. Heather, was that a question about the locus in terms of where the HBB gene is located? Is that?
So it was more that she was asking where you found that graphic.
Oh, I tried to cite it right there, so I'm not sure. Okay. I'm hoping that's where it goes. Yeah. Okay.
Okay, we'll be sharing the slides on the resource page afterwards, and so we'll get, people will have access to that.
Okay, sounds good. It looks like Dana found an article about the holiday junction, so thank you so much. Put that in there. Excellent. I love this crowdsourcing that's going on tonight. Okay, I'm kind of scrolling through. I know there's a lot of questions and comments over on the side. Oh, I do have one thing while I'm still waiting. If you guys have comments or questions, you can post them in there. I do want to, there's one thing that I did as a modification this year that with this kit that I think AP teachers would be really interested in knowing, is that I took tape, and you know how we, our students in AP need to calculate recombination frequencies and talk about linked genes. This is a great way to be able to do that. And to, I just put some tape on here. So, you know, we often use the fruit fly example of all the different genes that are on fruit flies and which ones are linked or not linked. And so I have like the body, wing, and eye genes for flies that are linked. And then, you know, we have the dominant versus the recessive version. And then you can kind of have students work through that to see like, well, if you did meiosis, what situations would you inherit those two genes together, or those two alleles together versus the crossing over that occur so that they get recombination. So that's something that I used this year, which was really helpful because then students could see how could you separate out linked genes, and that's going to be the crossing over that will allow you to do that.
We are back, and I think that we've gone to our old camera again instead of the new one. So we will, fingers crossed that we won't have any more technical problems tonight. Before I introduce Tim, I just wanted to answer another question that showed up in the chat, and that was about where our Science of Coronavirus videos are. And if you go to our webpage, on our homepage, there's a rotating feature, and one of those features is the coronavirus series. But you can also find them just by going to our YouTube channel. And there will be a link in the email that I send out on Friday for that. And there was another question that popped up about people may not be seeing all of the questions. So when you post in the chat, you can just post to the panelists, but then we're the only ones who see them. But you also have the option to post to all panelists and attendees. So we encourage you to post for everyone so that all the attendees will see that as well. Okay, so with that, I get to introduce Tim Herman again. And he is the founder for both the Center for Biomolecular Modeling and 3D Molecular Designs. I won't go into anything else, but I will just say that without his foresight and his innovative ideas, none of us would be here today in this webinar, or that either organization would exist. So with that, Tim's going to talk about ensuring genome stability while maintaining an escape hatch. And so as we were looking at this before, preparing for this tonight, we realized that really that session topic is probably a full 90-minute webinar in itself. So Tim may be just talking about maybe the first half of epigenetics, but we'll have a follow-up video available as well. So take it away, Tim.
Okay. All right. I hope that no one came to hear something about an escape hatch, because frankly, I don't know what that meant either. But what we were going to talk about, this was like months ago when we had a quick meeting to map out all these sessions. I was going to talk about DNA repair and how we wouldn't be here if we didn't have repair mechanisms, multiple redundant repair systems. But there's not time to do that and talk about epigenetics. So what I'm going to talk about tonight is epigenetics. And we actually recorded a little video yesterday. It turned out to be about a 22-minute video on epigenetics. And it turned out to be half of epigenetics, because epigenetics is complicated. So I don't think I'm going to show any slides. Well, can we show this first slide? Does the technology work here? We'll see.
We will.
I don't know. I've kind of changed my mind today what I want to talk about in the next 10 or 15 minutes. But since we have these slides, I'll start with them anyway. So I guess the first question I want to ask is down at the bottom of this slide, and that is, should you teach epigenetics? And first of all, I would back up and let's talk about genetics. And in a sense, that's what Chris was talking about. Chris was talking about classical Mendelian genetics. But I'm not even talking about genetics. When I say that a foundational concept of biology is genetics, yes, you need to know all of those things, meiosis, mitosis, that Chris was talking about. But for me, molecular genetics is where all of the excitement is. And more specifically than that flow of genetic information from DNA to RNA to proteins. So that's molecular genetics. And if there's one foundational concept that all high school kids should have a good understanding of when they leave high school, it's that molecular genetics. So epigenetics, should you teach epigenetics? And I would first say, remember, I've never spent a day in a high school biology classroom. So take my advice to you that I'm about to give with a little grain of salt. if I were teaching a freshman or some sort of introductory biology, I would not teach epigenetics because you run the very big risk of confusing kids. Complexity. You see this favorite poster over my shoulder here is life is complicated. So we love complexity. And I think science gets much more interesting when you begin to show students that science is not just simple concepts that we've simplified to make it easy to understand, but all of these seemingly simple things have levels and levels of complexity, and I love to get into those levels of complexity. Epigenetics will reveal levels of complexity that I don't think you should expose high school kids to. Perhaps in an AP biology, some sort of advanced honors biology, then you might teach epigenetics, but even at that point, realize that this is a emerging field right now. And the answers are not at all well known. So you run the risk, even in an advanced high school course, of going down a rabbit hole that your kids may not successfully climb out of. So I would first start by encourage you not to teach epigenetics. All right, now we're going to talk about epigenetics. Because if for no other reason, a lot of the things that we do in our summer courses is to provide teachers with ready-to-use materials that they can use with their students. But the other thing we do, we sometimes talk about topics that we have no intention of you teaching to your students. This is information solely for you, the teacher. I think it's really good for the teacher to know the subject they're teaching at a level that's a little deeper than what they commonly try to introduce their kids to. So I'm rambling, but epigenetics, it's simply the addition of these small chemical markers like methyl groups or acetate groups that are added to our chromatin. Now, you know, chromatin, what is chromatin? Well, chromatin is what you have when you take those condensed metaphase nucleosomes that Chris was showing you. And in all phases of the cell cycle, other than right there at the mitotic stage, those chromosomes kind of decondense and they become this sort of amorphous, dark colored material that makes up the nucleus of a eukaryotic cell. All right, so we're going to be adding small chemical markers to chromatin, and chromatin is made up of both DNA and histones. The DNA wraps around a complex of 8 histones known as histone octamer to make a nucleosome. So the point I want to make here is that in epigenetics, we modify both the DNA and the histones. And those are sort of two separate topics. So understand that if you're going to teach epigenetics, you have to decide which of those two kinds of epigenetics you're going to address first. You need to talk about both of them. And then the whole goal is in the end for you and your students to begin to get an understanding of how they talk back and forth to each other. That is, modifications to the DNA influence modifications that are made to the histone proteins. So epigenetics is a complicated topic. I think what I want to do, well, all right, so let's go a little bit further with a couple of slides. I want to start by showing you David Goodsell's landscape of the nucleus of a cell. And I think a lot of you are familiar with this image. This is part of David's tour of the human cell. So without going into that whole painting, I just want to show you this image. We're in the nucleus here. So way over here to the right, lost my mouse.
Go to the left.
There we go. Hope you can see my mouse moving around here. This green structure right here is the nuclear membrane, and you can't really see what it is because there's a big old nuclear pore complex here. But everything to the left of this green nuclear membrane, all this stuff in here in yellow and brown, this is in the nucleus. And in particular, here is a gene. Here's a double-stranded rope-like structure. This is the DNA. This happens to be a gene that's being actively transcribed by this RNA polymerase. But if you look for other yellow double-stranded DNA, you find it all out here, and then you see all of these funny little circular shapes. These are the nucleosomes. Okay? This nucleosome is the repeating structural unit of chromatin. And if these things condense during mitosis, you then get organized chromosomes that we can identify under a microscope. But during most of the life cycle of a cell, you have a bunch of nucleosomes in the nucleus. I'll draw one other thing to your attention because we're going to look at a graphic a little bit later that will come back to this. Notice all of these nucleosomes that David put right here, very close to this lamina, which lines the inside of the nuclear membrane. This is thought to be transcriptionally inactive or repressed chromatin. So the DNA you find in this chromatin is not expressed in this messenger RNA. Notice also these green proteins that extend from the nuclear membrane here to nucleosomes. There is a story here that I don't know, that we need to ask David to figure out what is he hinting at with these proteins here. Okay.
Did you point out the unbound histones?
Yeah. All right. So the other thing is, so where are the nucleosomes that previously bound up all of this double-stranded DNA? It looks like the nucleosomes that were on this gene have been disassembled. And if you look carefully, you see these little structures here. These are histone octamers. Here's a whole bunch of them here, here. So all over the place, you see indications of histone octamers that have been disassembled from this actively transcribed gene. So this is part of the complication you'll get into if you dig into current research on epigenetics, because many of the epigenetic markers that are added to these histones influence the disassembly and then the reassembly of these nucleosomes onto the DNA once this advancing RNA polymerase moves through the gene. So a very, very complicated topic. It's very, very interesting. If you have a lot of free time to read or watch videos, there's an amazing amount of information that's out there. I had the luxury of spending most of the morning watching videos, and there's really some amazing videos on the internet. And we'll put a link to at least one of those in the resources following this webinar. All right, could I have the next slide, Heather, just to remind me?
Yes.
Yeah, all right, so I want you to think about this. Chris was talking about the human genome, 3.2 billion base pairs of double-stranded DNA divided up into 23 chromosomes. But the other thing to keep in mind is that in those 3.2 billion base pairs distributed on 23 chromosomes, only 1.5% of that DNA is used to encode proteins. Only 1.5%. All right, so imagine then in the nucleus, we just have this image from David Goodsell where most of our DNA is wrapped up into nucleosomes. And that kind of makes sense because we only need 1.5% of it to be accessible to an RNA polymerase. But then how do we distinguish? How does the RNA polymerase find that 1.5% of the genome which actually encodes proteins? It gets even worse than that because you only express a subset of those 1.5% in any given cell type. You don't express the same genes in a liver cell as you express in an eye cell. So it's even more complicated. All right. So I want to give you all of that as additional levels of complexity that we commonly don't worry our students with, for good reason. But at the same time, remember, when you oversimplify something, it can almost become trivial, especially to your, shall we say, high achieving students who are ready for more. So as a teacher, you have to be really smart about who you're teaching, what they're ready for, but don't be afraid to, don't swamp them, don't confuse them, don't drive them away from science. But at the same time, be ready to give them something more if you think they're ready. Okay, so can we go to that graphic?
You don't want to do some models first? Here we go.
All right, am I running out of time already?
We should move on to models soon.
All right, I want to show you just one graphic here, which there will be a link to this graphic following the webinar. I like this because it shows you sort of the whole story here. So these are, of course, some nucleosomes, but this is sort of extended nucleosomes that are transcriptionally active, whereas most of the nucleosomes are densely packed like this. We give this a name, heterochromatin, which is unfortunate. This is heterochromatin, this is euchromatin. And, you know, I used to do research in this area and I still can't keep those two words straight. How do we expect students to know the difference between those two words? But I just want to point out what's going on here. If we zero in right here to the double-stranded DNA, there are two little brown dots here. This represents epigenetic markers added to cytosine. And that's what we're going to talk about with this DNA model here. So you modify both the DNA at cytosines with methyl groups. So these are methyl cytosines. And then also there are some epigenetic markers up here on the histones. And they can be methylated or they can be acetylated. And all of these green markers here, the easiest way to think about this is these are acetate groups that have somehow disentangled these nucleosomes and their associated DNA from this repressed chromatin. So this chromatin is now accessible to RNA polymerase, and they show the RNA transcripts on this DNA. For some reason, they don't show you the RNA polymerase, which is kind of strange. All right, so let's do this with models then. So if we can forget about the slides. I wish you all had this model of a nucleosome. So let me just tell you briefly. A nucleosome is about 145 base pairs of double-stranded DNA wrapped around 8 histones. There were 2 copies of each of the 4 core histones. And these are H2A, H2B, H3, H4. Don't worry your kids with what they're called, but it's a complex of histones. The DNA does not make 2 full turns around the histone core. It makes 1 3/4 turns. So this DNA ends here and it ends here. And you'll notice that the histones, especially this blue one here, this is histone H4, and it is present right here where this DNA ends. And this DNA goes off to another nucleosome here and off to another nucleosome there. And these histones are basic proteins. That means they have a lot of arginine and lysine side chains. And those are side chains that are positively charged. Okay, and the DNA, of course, is negatively charged. So it makes sense that if you want to package DNA up in a non-sequence specific way with these histones should be positively charged. So they're perfectly designed to bind in a non-specific way to the negatively charged DNA backbone. So the most common epigenetic marker that's added to these histones is an acetate group, and it modifies lysines. Lysines are normally positively charged, but when you add an acetate group onto that lysine side chain, it essentially neutralizes that charge. So that's the simplest way to think about the impact of an epigenetic marker added to histones, is you reduce the intrinsic positive charge of the histone that relaxes its grip on DNA. And therefore, this nucleosome is going to be able to sort of dissociate itself from neighboring nucleosomes. And it's going to become part of that looser, more accessible chromatin that now an RNA polymerase can come along, recognize the promoter region. And then in recent work that is fascinating, but you probably don't want to go there with your students, there are a whole bunch of other proteins then that are involved in further disassembling, destabilizing each nucleosome, so that as an RNA polymerase approaches this nucleosome, it will kind of disassemble, release histones. They'll be held over here, stored transiently until the RNA polymerase goes through. They can be then redeposited on the gene. All right, so that's nucleosomes. I'm gonna go from this very nice 3D printed model of a nucleosome to this model of a nucleosome. And this is just a little plastic solenoid, and it has some spikes on it. They look like vertebrae. I noticed that Chris used that same term. They do look like vertebrae, right? But what do you think these spikes are? What if I told you, if you had this in your hand and you counted them, you'd find 8 spikes. And you'd see that 2 of the spikes are bigger than the others, other 6. So those 8 spikes represent the N-terminal end of the histones, right? Okay, so we can take a piece of double-stranded DNA right here, and we can then wrap that double-stranded DNA 1 3/4 turns around this histone octamer. And what we end up with is a nucleosome. Okay. All right, and then we can put another one here. And the space between the nucleosomes are often known as linker DNA. And of course, this then is the first stage. These are the individual nucleosomes. They will go ahead and further condense in ways that people are still arguing about to produce these higher order, densely packed arrays near the nuclear membrane, which are inaccessible to polymerases. But then if you acetylate the histones, you can disentangle this so it's more accessible. I want to turn now to the main thing I was intending to talk about, and that is you can also modify the DNA. So let's turn to our DNA model. Let's turn to this DNA model first. This is our simplest DNA model. All right, and here's just a sequence. And what I need to just plain tell you is that we modify cytosines, blue cytosines, with the methyl group. We only do that if the C cytosine is followed by a G. And therefore, you'll read about CpG sequences. So there's a cytosine, and then there's a sugar phosphate that joins this cytosine to the next base, which has to be a guanine. If you find a CpG, we have a whole bunch of enzymes that will recognize that CpG, and it'll put a methyl group on that cytosine. And what is really interesting to me is that if you have a CpG in this strand, you'll also have a CpG on the other strand. And of course, when I say CpG, I'm going from the 5' to the C-terminal end, 5' to 3' direction. So this CpG will be pointed in the other direction. So these things are really hard to see in, just a sort of a random sequence of DNA. But the point is, whenever you have a CpG in a human genome, you have the chance of adding a methyl group to it. And I found an interesting number even today. I've been wondering about this for some time. and I found the number, I assume it was right. It said that about 70% of all of our CpGs are methylated. So this is not a rare epigenetic modification. 70% of the time we have a methyl group on those CpGs. All right. This gets even more interesting if you look in even more detail at the molecular level. Heather's trying to anticipate what slide
You're going all out of order here.
I know. We usually practice the order, you know, very well, and I'm
We're supposed to
irritating
be able
her.
to switch back and forth easier.
I know. It's all because I watched those videos this morning. All right. I want to show you a GC base pair. So here's the C. I want you to imagine it is followed by a G. And I want to point out which atom on the cytosine is methylated. And the answer is it's the number 5 carbon. So we methylate the number 5 carbon. So we might represent that by taking one of these hydrophobic yellow clips, because a methyl group is hydrophobic. So I'm just going to put this yellow clip on the number 5 carbon of cytosine. Slide up. Yeah. Okay, now we're going to go over here to our dynamic DNA model, and I want to show you where this number 5 carbon is in double-stranded DNA. All right, so I'm going to lay this down. Can we go back to the overhead camera?
Okay.
Because
Hold on a second. We can
yeah,
zoom.
all right. Now, first of all, just look at that for a minute. And no wonder kids get confused and annoyed. Look at the color there. And, you know, I'm just looking at this for the first time now, and look how bright these yellow phosphorus atoms are. So I want to call your attention to these bright yellow methyl groups I put in here. And yet we've got all this other color going on. And in this model in front of me, trust me, these phosphates are orange. I don't know why they're showing up as such bright yellow ones here. But the point I want to make, and I don't know how well this is making it. I think, Heather, I want to turn this back up this way, because I just don't like that image very much. The point is, those number 5 carbon of cytosines are directed right into the major groove of DNA. They're not around behind here in the minor groove. The number 5 carbon will be in the major groove. So when you add a methyl group onto that number 5 carbon, the methyl group is hydrophobic, right? And we also know that proteins which bind to double-stranded DNA in a sequence-specific manner, these so-called transcription factors, they're looking for a pattern of hydrogen bond donors and acceptors and hydrophobic methyl groups in the major groove of DNA. So the epigenetic markers that we make, the methyl groups that we add to cytosines, are right there in the major groove of double-stranded DNA. Okay, and now I want to go back to this slide and show you one other thing. Here's
Okay, give me a second.
okay, there's a slide coming up here. I've just made up a sequence here. Lost my mouse again. There we go. I've made up a sequence and I've colored the cytosines in blue. And you'll notice going 5' to 3' on the top strand, here's a C followed by a G. So this is a CpG sequence that we could methylate. And if we go down here, here's another CpG. These C's before, we can't methylate them because they're not followed by a G. But if we methylate this C on this strand, and it's sort of, dare I say, homologous C on the other strand. That's not the correct use of the word homologous, but if you methylate it, that's what you get. You get 2 methyl groups, and I just told you those methyl groups are now going to be sticking right out into the major groove of DNA. So any protein that was going to bind there in a sequence-specific way is now going to have to take note of the fact that, oh, there are 4 more methyl groups close together in this major groove of this DNA sequence. But here's the kicker. You see these T's, these thymines? Here's where you need to know this. You need to be able to draw the chemical structure of all these bases. And tell you need to be able to do that. I can't do that. I have to review every time that I need to do it. But you need to have these structures nearby. You need to show them to your students, because every one of those T's, those thymines, already has a methyl group on its number 5 carbon.
Let's show it here, and then we can do it in that other model.
Okay,
right?
so I'm going to click here. There are all of the methyl groups that normally exist all the time. They're not epigenetically added. That's just the structure of a thymine. So you have all those other methyl groups. So it's not like a transcription factor isn't used to seeing methyl groups. It sees methyl groups all the time. Every time there's a T, here's a T right up here. That little gray carbon represents the methyl group. But the only point I'm trying to make is that the methylation of CpGs in double-stranded DNA is a really significant modification. And here's something I really like about the double-stranded or the dynamic DNA. I've taken the same section of this DNA model where I have a C followed by a G. And I can take that now and I can unwind it and I can lay it down on the table in front of me. And suddenly what I've been trying to tell you might make a little more sense. Because first of all, you can easily see the 5' end to the 3' end. So looking down here, I can examine each of these bases and I can see that this is a C followed by a G. Therefore, I can put a methyl group on that 5 carbon of this C. At the same time then, there's a corresponding C on the other strand, 5' to 3'. There's a CpG, and therefore I can add, and I shouldn't say I can add, I should say there is a methyltransferase. There's an enzyme, a methyltransferase that will recognize this CpG. It will actually flip out, it'll break this base pair, it'll flip out this cytosine where the enzyme active site, the methylase active site, can get a hold of it and actually enzymatically add that methyl group. And of course, there's a 3D structure of that methylase with a cytosine bound in the active site. And that's a structural story that is well known. All right, so that's about all the further we can go in this short, 10, 15 minutes. It's an introduction to epigenetics. It's a fascinating topic of current research. There are many human diseases that people now are convinced there's an epigenetic basis to them. One of the, we'll put a link to 2 videos associated with this topic on our website. And parts of those videos you might want to show to your students. But if you can find some time, and I know that's kind of unlikely, but if you can find a time to watch these, they're really well done. There are two women do these videos. Outstanding people. Carolyn Luger is a researcher who does this work. Carolyn Luger's group was the group that actually first came out with the first high resolution structure of the nucleosome by X-ray diffraction. And of course now she set up a big cryo-EM lab where they're continuing to unravel this story of epigenetics and all these other proteins that bind to nucleosomes. Epigenetics is fascinating. Be careful. Lots of rabbit holes out there. If you have time to explore rabbit holes, there are some fascinating ones. but not for the faint of heart.
Okay, thank you.
All right.
Please post your questions in the Q&A. There have been a lot of comments and maybe some questions in chat, but I've lost them. So if you want us to address things, put them in the Q&A. And we just want to go ahead and wrap up tonight to make sure you guys know what we use. So Chris Chow used our chromosome connections kit and the chromosome student modeling pack. We also have a combo pack where you can get, where we combine the nucleotides and the chromosome. So, and then Tim used our dynamic DNA kit. We also have a nucleosome mini model. So unfortunately, this big one that we use is a nylon one. We'll pull out that mini model that we have that's available so you guys can see that.
This is a beautiful model. If you're holding it in your hand right here, 4 inches from your eyeballs.
You can't really see a lot with it, but you do need to hold it.
You really can, but it's not to hold up in front of a group like this trying to impress them.
So our 20% off all of our regular kits is still going on. And then starting tomorrow, we have our stock up sale on chromosome kits. So if you buy 4 or more of the chromosome connections kits, you'll get 25% off. Next week, we actually have a break. And then in 2 weeks, Tim is coming back and doing genome editing, where we are going to be focusing on the CRISPR, the adaptive immune system. And then please remember to complete that form for the certificate of participation and the survey as you leave. So thank you, and let's try to address some of the questions.
Yeah, I see one question there I really want to answer. Gabriel asks something about, when there are addition of methyl groups, does that attract transcription factors? And the answer is yes. So you're getting exactly the right idea, that it attracts either repressors or activators. Generically, in the field of epigenetics, you talk about writers, enzymes that write epigenetic markers, or erasers. So you have methylases and you have demethylases. And then you have a third class of proteins called readers. These are proteins that just recognize the presence or absence of those epigenetic markers, and they bind or don't bind. So that's exactly right. And that's why it gets complicated, right? Because the methylation of CPGs in DNA, and by the way, those CPGs are something called CPG islands. There are regions of our genome, usually located near or in promoter regions, where you have a large clustering of CPGs. And therefore, you have the potential to put lots and lots and lots of methyl groups in a promoter region. And again, without getting too specific about it, I will say that it either attracts transcription factors which bind to it because it's methylated, or they may prevent an activator, another transcription factor that's an activator of that gene. But with the epigenetic marker there, you may prevent the normal expression of that gene. The other thing I was going to say about epigenetics and human disease, if you do a Google search for DNA methylation, you'll find references to papers that are talking about hypermethylation in Alzheimer's or other dementias. So there are research groups out there that are very actively identifying and documenting the epigenetic markers around genes that are known to be involved in some of these neurodegenerative diseases and cancer. All right.
Okay, I'm going to interrupt you for a minute. There have been a couple of questions that I'm going to answer. So one, our stock up sales, each stock up sale goes for 6 weeks. The 20% off with the webinar series, that goes until the end of June. And somebody was asking about our orange nucleosome kit. So this is still in a field test. And so maybe in a year or so, we might have these available, but I have good news for you guys. The CRISPR kits that Tim's going to be highlighting in the next 2 sessions, those are going to be available for sale in March, and hopefully we'll be able to ship in April. It's going to be real tight right now, and I'm figuring out the pricing on those actually next week. So we will, those will start to be available for the general public.
I just want to show one quick thing. See this nucleosome? I said that if you acetylated histone H4, see these little red caps? These represent acetate groups, which cancel out that positive charge, which now means that this nucleosome is going to lose its grip a little bit on the DNA that was formerly wrapped up in this nucleosome. That opens or disassembles this nucleosome to the point where you can now transcribe it. And it doesn't do so by dropping out a whole histone octamer. It's a much deeper rabbit hole than that, but at least you can acetylate the N-terminal tails of histones in these nucleosome models.
And we are so happy that we're able to use these clips from our amino acid starter kit, and they work with our dynamic DNA and eventually the nucleosome kit to represent some things. So I saw another question here from Gabrielle about data that the Goodsell paintings are based on. You want to?
Yeah, so David is a very meticulous guy, and that's why I'm embarrassed to say, I've been looking at these things for years and years and years, and I just learned something this week about those histone octamers that he scatters along that gene that's being transcribed. So to answer your question, he has a book called The Machinery of Life, and most of the posters that we originally put together for these landscapes came out of that book. And for each one of those early landscapes, David published a paper in which he gave the information that you're asking for. So if you send us an email, ask us this, we can provide to you a PDF of the paper that David published in which he talked about why he illustrated things in various ways, and even gives references to the literature where he sort of documented what was known about that particular protein in his landscape.
We'll add that file to the webinar resources page.
Yeah, we can certainly do it for the tour of the human cell. That was one of the first landscapes that he published a paper describing. But his other landscapes have other papers associated with them.
Okay.
So I think it's also good to note that science being science, that he painted the tour of the human cell several years ago. And so there may be new information now available that would not correlate with what is in the painting.
We like to say that all models are wrong. And I would never criticize a Goodsell landscape because, again, David he put so much into those landscapes. But based on the videos I watched this morning, the way in which he has represented that transcriptionally active gene as being completely devoid of nucleosomes, and yes, he put the histone octamers scattered around that naked gene. But what people now know now is that the disassembly of those nucleosomes occurs in kind of a stepwise fashion where the H3, H4 tetramer the H2A, H2B dimers are kicked off first by a big protein complex that kind of binds to and disassembles this nucleosome. You'll never guess what they call the protein that does that. There's a protein called FACT, F-A-C-T. And you know what FACT stands for? The facilitator of all chromosomal translocations. I'm not kidding you. And they have the 3D structure of this huge heterodimer that sits right on top of a nucleosome and senses an approaching RNA polymerase. And I'm just making this stuff up now. I haven't read these papers in detail, but it somehow facilitates the disassembly of a nucleosome so that the RNA polymerase can in fact run through that DNA that was formerly wrapped around a histone octamer. Fascinating stuff.